This is a quick demo of the Strandomics system, which we use at Strand Life Sciences to do all our reporting of clinical tests. We run a fairly large number of clinical tests spread over rare diseases and somatic cancer. And this demo will cover the somatic cancer piece. So here's a case, and you can see in this case, this is a 54-year-old male with colorectal cancer. The TRFs and all of the lab-related QC pieces and results are already archived as part of this case, so that everything is in one place. And the variants for this case have already been fed into the system. Our variant calling happens in a separate platform called the Strand NGS platform, which has pipelines for quite elaborate pipelines for all kinds of variants, including structural variants and copy number variants, and those are already fed into the Strandomics system. So now when we open this case, you'll see it opens up with 4 variants, and it's in the standard of care tab. The standard of care tab talks about variants that influence um, drugs that are approved for this particular tumor type, which is the colorectal cancer tumor type. So drugs that would be normally used as part of NCCN guidelines for treatment of colorectal cancer. Any mutations that impact or modulate these drugs are the ones that are mentioned here. There'll be other mutations that are found. The panel that we ran here is a 152 gene panel, so clearly there are a fair number of genes, so you'll get many more mutations as well, somatic mutations. And those will impact drugs that are not approved for colorectal cancer but might be approved for other tumor types or might be in trials. And we'll get to those as we get to the investigational section. But just focusing on the standard of care section right now, there are 4 variants. These have been automatically annotated, as you can see, with a variety of different annotations. You see the quality annotations here. All green means that the quality is good. These include base quality, mapping quality, and a number of other quality indicators. Um, One of these is a deletion. 3 of these are point variants. The deletion, and you can interpreters actually go over each of these and pick which ones they want to report. So in this case, they've chosen not to report the deletion. So let's go a little deeper into the deletion and see why. Um, if you look at the deletion, this is the visualization for the copy number. So copy numbers are called against a backbone or a profile, and this is the for every exon. So there are 11 exons in this gene, and the dots determine where the current sample is relative to that backbone. So there's only 2 exons where there seems to be a notable deletion, and even that is within the range of error. So it's for that reason, it's not a very strong deletion. Now, of course, at the variant calling stage itself, one could have done this, but our approach has been to feed more variants, be conservative at that stage, or be liberal if you want at that stage, and feed more variants into the interpretation pipeline and let the interpreters actually pick and discard variants as needed. So this variant looks weak, so it's therefore been not considered. All the other 3 are actually strong, and we could get into those. For instance, the KRAS variant here, and it's very good quality. It's found in Cosmic, quite a lot of occurrences in Cosmic. The domains that it's carried in, and we've curated all of this, the domain information. It's in an important domain, and since it looks important, the reporting text for this variant has also been curated, and you could of course edit it, but if it's been curated once, it's pre-populated here. So once you go through these variants, 4 variants, and you're sure that these look good, the 3 that you want to report, the 4th you've eliminated, the 3 that you want to report are good, then you can go to the interactive report section, and it'll automatically generate a report from these chosen variants. Now, an interesting thing about this report is that it's a drug-based report. It lists out drugs that are part of the standard regimen of treatment for colorectal cancer, and for each drug, which are the markers that were tested as part of this panel, which markers and mutations were actually found, and these were the ones that we picked on the previous page. And then accordingly, what was the predicted response? Is there increased sensitivity? Is there increased resistance? And all of this is automatically done. And there's a table at the bottom for toxicity as well. So there are a few toxicity markers. In this particular case, we didn't find any. But if we did find something, then that would be listed here automatically, and the relevance would also be found automatically. So the way this whole table is derived completely automatically is driven by a lot of curation and rules. So to show you those rules, we can go to um, the standard of care section out here, and I've got it out here. So let's go to the colorectal cancer section of the standard of care. And in this section, lists out all of the markers that influence standard of care treatment for colorectal cancer. And so these are the genes involved, and then these are the drugs involved and their dependency on the genes. And as you can see, it's quite elaborate. There are lots of drugs involved and their dependency. This is some logic rules that have been captured. And with each logic rule, there are evidence levels. And if you click on that, you will see the actual curation that's been done, the actual text that will show up in the reports. This is clinical evidence, and we also have preclinical evidence. And what you see in these double brackets is a PubMed ID. So once you put it there, the report generating mechanism will automatically expand it out into a full reference. You see these various levels of evidence. These are levels of how important is the evidence associated with this rule on the impact on this drug. So we only consider, we have a certain classification scheme, and we only consider high quality evidence, and within that high quality, it's graded as 1A, 1B, and 2A. So when you clicked on the interactive report, after we selected the 3 variants that we wanted to report on, it automatically generated this report based on all of these rules. So that's what we call the standard of care report. And the reason we've automated it entirely is that this report goes within a week of the sample arriving in our labs, and so therefore time is short, and so we've completely automated this part. You could now go back and look at the investigational section. So if we click on the investigational section, it's opened up here. And in the investigational section, of course, you have a lot more variants that need to be looked at. And as you can see, these variants have been automatically annotated with various, okay. So they've been automatically annotated as gain of function or loss of function with a confidence level associated with these. And our interpreters would now go through these and pick which variants to report on, and typically they might choose some filters here. For instance, let's say you want to start by looking at point mutations, and you only want to look at things which are gain of function or loss of function. Everything else is either an ambiguous category or an unknown category that you might not want to report on. So when you apply this, you get a handful of variants. You get about 17 point variants that need to be looked through. And for each point variant, you've got all of the quality indicators, you've got the supporting read percentages, and together based on that, our interpreters can very quickly go through this and decide which ones to report on. For instance, this is all green, so that's good quality. There are various shades of red here, and so unless a gene sounds particularly interesting, our interpreters will probably hit the cross button here. And you can also see that there are supporting read percentages here. So if something is like 2%, then you might hit the cross and give a reason here saying low supporting reads 2%, hence need not be reported. So this is a relatively quick process, and some of the other things that are completely green, there might be other reasons to not report them, and those are mentioned here. So just by going through this, the variants that need to be reported are picked up. And so those were the point variants, and now one could do the same with amplifications and deletions. So you could pick copy number variants. Okay, so we've got a bunch of copy number variants here, and let's take the first one. It's in the ORCA gene. If we zoom into that, We can look at the copy number profile to verify that the copy number looks consistently above the interquartile range in the backbone profile. The copy number visualization. This is in chromosome 20, so if you go to chromosome 20 out here, so you can see the ORCA gene and all of the exons are consistently amplified to a copy number 3 level. So this is copy number 3. So all of the exons are copy number 3 or above. So there seems to be a good amplification there. The report, accordingly, you can write the reporting text, and if it's written once, then it's auto-populated that this is the reporting text that goes in for this variant. That describes that amplification is correlated to overexpression, and that overexpression has been reported to lead to carcinogenesis. This is still not about drugs, it's just about the fact that the amplification causes carcinogenesis, that this variant is indeed carcinogenic and ought to be reported. Okay, so similarly, once you've gone through these other variants, one can write reasons why one has put a cross or a tick. So for instance, FLT3 expression is almost exclusively reported in heme cancer, so not in colorectal, so it's a cross has been put. So one goes through these and puts ticks or crosses, and once the relevant variants are selected, you go to the selected variants tab to now identify drugs that need to be reported. So in this case, we have only 3 selected variants. Let's take the amplification that we looked at, the ORCA amplification, and now how do we pick the drugs that need to be reported? So automatically the system will identify all the drugs that are in pathways, drugs that are either direct targets of ORCA, that directly target ORCA, or drugs that target genes that are upstream or downstream of ORCA. And now our interpreters go through this list and decide which of these drugs to report on, and some knowledge of cancer biology and the pathways is needed here. And they use that to determine whether to pick any of these other target genes or not, or just focus on the direct targets. So in this case, they focused on the direct targets, and they picked the first drug that's applicable, and hit a cross on the others. And they've hit a cross because the clinical trials are not relevant, etc. So let's go into the first one here. And so here is where the evidence of why this drug is applicable to this patient is written. So there are, as you can see, there are 4 sections here. This is the summary section that goes into the report saying amplification of ORCA has been reported to show positive correlation with expression and malignant transformation. And then for this drug, there's clinical evidence that gets listed here and preclinical evidence that gets listed here. So in this case, there is no clinical evidence, and preclinical evidence is also missing, so we just put a sentence saying there isn't any. So now we have to remember that clinical and preclinical evidence is tumor type specific, so there is no evidence in colorectal cancer. But now, is there evidence in other cancers? So you could choose here. We have curation done for several other cancers, so if you click on this, it'll say choose from best others, which means the other tissues, the other tumor types, do we have those curated? So breast cancer, we have a fair amount of curation, there's preclinical evidence there. Then there's gastrointestinal, there's preclinical evidence. There's breast cancer. So all of these have been already curated. Different ovarian cancer, there is some evidence. So you could pick across all of these and then write a global summary that's pan tumor type, saying preclinical studies from multiple cell lines harboring ORCA amplifications, including gastric, breast, ovarian, have demonstrated treatment benefit in response to this drug. So whenever there isn't curated information or information literature available for a particular tissue, you pick from other tissues and you take this choose from best others tab. Or you could write a more general statement for the entire drug class, not for the specific drug. So for instance, for this drug class, there are other inhibitors that have been shown to result in cell cycle inhibition. And so you could write a general statement, and when you take this statement will also be copied into the report. So now you're ready, you've got everything filled in, and if this was already filled in earlier, then it'll be auto-populated. And if you look at the total amount of curation that needs to be done this way, you've got genes, a number of genes and tissues, so it's the two-dimensional combination, gene-tissue combination, for which you've got to curate this information. And our estimate is that there are about 300 odd gene-tissue combinations which are very commonly found, of which the most frequently occurring 150 or so have been completely curated in the system, and the remaining are being done on the fly basis as new cases come in. So now we are all set. Once we've picked our variants, we've picked the drugs to report, um, and I also forgot to show you there are clinical trials that are automatically picked up by the system. So all of those are listed here, and our interpreters can go through each and decide if they want to pick some specifically. So there are trials for the same cancer, and there are trials for other related cancers. So in this case, they picked the two trials that are available for the same cancer. One of them is recruiting, one of them is active but not recruiting. Of course, you could choose to pick others as well. And now you're all ready to report, and all you now need to do to report is to click this button, View Reports, and the entire report will get generated automatically for you. Let's see what the report looks like. And here is the report. It's got, in this case, since I do not have appropriate role permissions, the patient names and demographics are all blinded, but if the report sign-off person were to come in, he would see all of these things in the report. The indication, the standard of care report, which we talked about earlier, the drugs that are specifically relevant, specifically approved and used for colorectal cancer. And then this is the investigational section, the drugs that were picked up, and the trials, and which marker they were based on. So that's the front page, as easy as that. One look at the standard of care drugs, and one look at what repurposing or clinical trial possibilities are being offered. And then for every one of these drugs, we have a full page, oxaliplatin, there's a full page here describing all of the evidence that was filled into the text boxes, and it's just automatically copied in here, along with the references, which we put in double brackets in those text boxes, and that's all those are automatically obtained here. So we are set, the report is ready. One page for every drug, it's neatly organized. If you look at each drug, it's neatly organized into clinical evidence, preclinical evidence, the information about the mutation itself, and the clinical trials, that and a description of the drug and references. And at the very end, we have a list of regions of the targeted regions that are not quite adequately covered to the tune that one might want. So this is a disclaimer saying these regions are not are probably not as well covered, just for the record. And then all of the supplementary information on what genes were tested, etc., etc. All of that just automatically shows in. And we generate almost 250 reports a month using this system of the nature that you've seen now. Thank you very much.